Good morning. My name is Sara Montanari. I'm a postdoc in David Neal Lab working on paired genomics. And as a tree geneticist, I'm happy to introduce Sean Myles. Sean had a pretty interesting career. He started working on human, and then for his postdoc, he moved to grape. And today, he's associate professor at Dalhousie University in Nova Scotia, where he's working on apple. There, he established an apple biodiversity collection and is doing genotype to phenotype association. And today, he's going to talk about mapping genes in apple and grape. Welcome, Sean Myles.
Okay. Yep, sure. Great. Thank you very much for having me. On Thursday, when I woke up in the morning to come on this trip, there was a little bit of snow falling in Nova Scotia, and it was very cold. I noticed on the map you just showed there that Nova Scotia actually got cut off the map of North America. So for those of you who don't know where it is, it's east, and we're a little peninsula hanging off the edge of North America there. We're not Newfoundland. That's the rock out in the middle of the ocean, but so we're not quite that far out. But thank you very much for having me. This is a wonderful place, cycling around at this time of year. I know some people are like, ooh, it was cold this morning. It wasn't that cold this morning for me. I thought that was wonderful. I have a sabbatical coming up starting on July 1st for a year, and I'm thinking Davis sounds like a really nice place to go. So if anybody has any room in their lab and they're looking for somebody to hang out, I might be interested. So let's get started. The first thing I wanted to mention is that a lot of the work I'm going to be presenting today was done by Zoe and Kendra in my lab that are just about to defend their PhD theses. And so I just want to acknowledge them to emphasize that they did most of the work. Michael actually didn't do any of the work, but he's a postdoc in the lab. He works on chocolate and marijuana, hence the big smile on his face. So, but I'm going to talk about grapes and apples today. So a lot of what I'm going to keep it very simple on the genetic side. We're going to talk about SNPs and only SNPs, and some people are like, oh, gee, they keep talking about these SNP things. Let's go over this quickly. So it's the most common form of genetic variation available, and as a geneticist, what we want to do is find those genetic differences and correlate them with phenotypes, of course. And so if we were to look at a couple people in the room here, and I take my sequence and I line it up with your sequence, and then we start on chromosome 1 at the very first position, and we walk along the genome, then generally the book looks very much the same between you and me, and we only see a difference every once in a while. And it's these differences that we're interested in, not the invariant pieces, but the variant pieces of the genome. And as we walk across individuals, eventually we usually find somebody where we have one nucleotide difference here. It doesn't quite line up right for some reason. But what we see here is this individual carries a T, while everyone else carries a C, and that's a single nucleotide polymorphism or a SNP. And so as a geneticist, what we really want to go and acquire is genome-wide SNP data. We want to be able to take a set of samples and start at the beginning of the genome, walk through the whole thing, sequence the entire thing, and determine where these are, where these SNPs are in the genome. So it sounds pretty straightforward. In theory, we would just go through and find all the positions that vary, and then we would have genotype data for all of your samples. In practice, of course, it's a lot more difficult, and there's probably students sitting here right now thinking, yeah, I've got these FASTQ files and I don't know what to do with them. And it does end up being a little bit trickier than that. But in the end, I'm not going to go over the details of how you get these data, just needless to say that there were two methods used that I'm going to talk about today. The data that we'd use from grape came from the Vitis 9K SNP array. That's a genotyping microarray, and that we developed as part of my postdoc project. And then more recently, we're using ES genotyping by sequencing on apples. So at the 30,000 foot level, if you're going in to start genotyping a whole bunch of long-lived woody perennials and you work on almonds or walnuts or whatever it happens to be, don't make an array. Just use next-gen sequencing. There's lots of problems that we've published on about probe sequence hybridization in these high-diversity species. It's a real pain to work with. The data are really, really terrible. And you have a lot better chances here if you just jump straight to the next gen. That would be the piece of advice at that level. But in any case, what you want to end up with is you go through the genome, and for each SNP, you can see as a column here in a table, you want a table of genotype data. That's your aim. And in the end, you have a table of genotype data that looks something like this, where you have your SNPs as columns, your samples as rows. Individual 1 here is AA homozygous, sample number 2 is AT heterozygous, this sample is TT homozygous, and so on. And of course, you're going to have some missing data. That always happens. You end up with a table like this. And what you want to be able to do, if you think about it, you want to convert this table into a series of zeros and ones. Once you have that, zeros, ones, and twos. So, and for SNP number 1 here, if you take it and you say, how many T alleles do these individuals carry? You either carry 0, you carry 1, or you carry 2. And for each SNP, you just count up how many of that alternative allele do you actually carry. And so now you have a big table of zeros, ones, and twos, and you can ask really simple questions like, how closely related is one sample to another? Well, how many times do they share a 0? How many times do they share a 1? How many times do they share a 2? And so on across the genome. Makes it very simple. So what you're aiming for is this kind of numeric matrix of zeros, ones, and twos that you can use to describe the relationships among the samples. Unfortunately, you're going to have some of that missing data there, and that's one of the problems that we've worked on a lot. In working with non-model organisms that have really crappy reference genomes, where things are not really well put together. In the apple, for instance, only 70% of the genome is actually put together, and a lot of it is not put together properly. And so you don't want to rely on physical information when filling in that missing data through imputation. And so we've developed some algorithms in-house, one called link impute, where you don't need a reference genome, and you can still impute the data. It performs really well. And we've got a better version of that available at our website called link impute R that goes straight from the VCF file and uses the raw recount data if you're interested in checking that out, and that'll be published soon. So once you get that full table, it's completely complete now because you've imputed your data, and you've got zeros, ones, and twos all the way across. And then you've got some phenotype data for your individual. Something ranges from 1.4 to 8.2 and so on. Genome-wide association studies, or GWAS, and genetic mapping, association mapping, is simply taking each of these columns here and correlating it with your phenotype. And then do it again for SNP number 2, and SNP number 3, and SNP number 4, and in that manner, you collect a bunch of p-values for this correlation. You have 100,000 SNPs, you have 100,000 p-values, you look for the lowest ones, and you say, does that make any sense? Might that be a region of the genome associated with the phenotype that's biologically meaningful? And that's basically it. Now, the correlation isn't just a standard Pearson correlation. You have to correct for a few things, but that's the general idea. Once you get your table of zeros, ones, and twos, so all I'm going to talk about from now on is using a table of zeros, ones, and twos from apples and from grapes and seeing what you can do with that. So let's move on to one of many people's favorite organism, grapes. This project started during my postdoc in Ed Buckler's lab at Cornell when I came down here. Just outside of Davis, they have the USDA grape germplasm collection out in Winters. And it's a beautiful collection. It's got lots of a tremendous diversity of grape varieties. Today, I'm only going to be talking about variation within Vitis vinifera, the domesticated grape. When you go to the grocery store and you see table grapes and you go to the wine section and you see your Pinot Noir and your Cabernet Sauvignon and so on, those all belong to Vitis vinifera, the domesticated grape. There are 60 interfertile species of the genus Vitis distributed all across the northern hemisphere. Today, I'm only going to talk about one. And unfortunately, what crosses your palate is often just a very small fraction of the total diversity out there in the genus. But for the purposes of this study, we're looking exclusively within Vitis vinifera. And despite only looking within vinifera, there's this tremendous amount of phenotypic diversity in cluster density and size and shape of the berries and color and so on. And really fascinating. What stems from that, and that's all a result of this tremendous amount of genetic diversity. So coming from a human genetic background, it was very surprising to me to see that when you're sequencing, if you look at the number of nucleotide differences between Pinot Noir and Cabernet Sauvignon are on the order of the kinds of differences you would see between humans and chimpanzees, right? So what you see in intraspecific variation in these plants is often equivalent to the kinds of levels of interspecific variation you see in mammals. And so when someone from the mammalian world tells you that the SNP array is going to work really well in your species and you're working on plants, then yeah, you have good reason to believe that that might not be the case, because you're just dealing with levels of nucleotide diversity that are much higher. So that's one of the challenges in dealing with these long-lived woody perennial fruit crops. I'm going to point out a couple of more of them that would lead us to believe that it might be really difficult to do genetic mapping. And the next one is this rapid LD decay. So it turns out that grapes and apples have these very large effective population sizes for very long periods of time that have enabled recombination to break the genome up into tiny little chunks. They also didn't have a really very strong population bottleneck, and there hasn't really been that many generations since domestication, since they can be vegetatively propagated. And for all those reasons, LD decays really rapidly in the genome. So what does rapid LD decay mean? It just means that every SNP that you're looking at that you genotype is found on a chunk of chromosome. Essentially, it's tagging that area. And the area that it tags depends on the length of LD. So and the length of LD is determined because the pieces of chromosome travel through time and space as little chunks. And the chunks are very, very small in grape and apple, whereas in other organisms they can be rather large. So this is a double-edged sword for geneticists, because if the chunks are very small, then if you map something to that location, you're pretty sure that the causal locus is very close by. But in order to map it successfully, you're going to need on the order of millions of SNPs. So we find that LD is breaking down to, it's tough to find 2 SNPs within 500 base pairs in grape that actually have an R-square greater than 0.8. And the same in apple. It's like the trees that people study, like poplar and those sorts of things, where you see LD break down really, really rapidly, suggests that we're going to need millions of SNPs to do a well-powered GWAS. Well, I just mentioned the array that we're using in grape has a Vitis 9K SNP array. That means 9,000 SNPs. And then by the time you filter out the junk, you've got about 6,000 SNPs. And then throw out the minor allele frequency below a certain threshold, you got 4,000 SNPs. So we're woefully underpowered to be doing to be doing a good GWAS here, considering that we estimate that you're going to need millions of SNPs in order to do a good job. The other problem or potential challenge in doing genetic mapping of this nature in these crops is the fact that there's this intense relatedness among individuals. So from the grape genotype data, we can actually infer who has first-degree relationships with one another, and we can represent these varieties as nodes in a network, with the lines between the nodes representing first-degree relationships, where solid lines are parent-offspring and dotted lines are sibling relationships. And so if you start with Merlot down here in the corner, you'll see that Merlot has a first-degree relationship with Cabernet Franc that is a parent of Cabernet Sauvignon, whose other parent is Sauvignon Blanc, that is related to Traminer, who's a first-degree relative of Pinot Noir, which is a first-degree relative of Chardonnay. So when you're going to the store and you're looking at these grape varieties on the shelf and you're buying a bottle of wine, you're looking at this sort of family tree. Essentially a bunch of varieties that are all interconnected by a series of first-degree relationships. In fact, 75% of the varieties in the USDA collection here of the vinifera have at least one first-degree relative in the collection. Okay? So it doesn't mean that they're inbred. It just means that they're still highly heterozygous, and they've been vegetatively propagated for a long time. Pinot Noir is 1,000 years old. We've been vegetatively propagating it for 1,000 years. And so, which is a problem in and of itself that I'm not going to talk about today, but the issue is that it's all tied up, all that diversity is tied up in this sort of family network. And that high degree of relatedness among individuals can make some downstream analyses tricky for association mapping. At a higher level, in terms of population structure, what do we see when we take the genetic data and we want to look at the relationships among the samples that we've collected? We have about 6,000 SNPs and 600 varieties of grapes, and we can't visualize things in 6,000 dimensional space, so we use principal components analysis to reduce that down to 2 dimensions. And so what we're looking at here, principal components analysis gives us the ability to see what the relationships are among these individuals in 2 dimensions. We have the primary axis of genetic variation, PC1, explaining 7.6% of the variation in the data. And you see here that in general, the red dots are found on this left side and the blue dots over here. The red dots represent Western grapes that would be sort of Germany and France and Spain and so on. And they tend to be wine grapes. You can see that they're mostly circles over here on this side. Whereas over to the right, you've got primarily triangles that are blue, which are Eastern table grapes, tend to be from the Middle East in this collection. And so that's unsurprising that the primary axis of variation sort of correlates with geography. That's often the case with a lot of crops, and also with use. But that's also correlated with religion. You know, Islam has been the predominant religion in the East, where the Eastern grapes are from here. And since the 6th century, alcohol consumption has been prohibited. And so the main target, and they're selecting for table grape attributes and generating table grapes in that region, whereas in Christianity in the West, where alcohol consumption was not prohibited, the target is to have better wine grapes, which results in very different qualities, very different breeding targets, obviously, and different use. So you've got geography, use, and religion all kind of tied up along that primary axis. I don't know how to disentangle them and see their individual effects on the genome. Would be really interesting if anyone has any ideas. But in general, this only really becomes problematic when you realize that table and wine grapes also differ according to some of the phenotypes that we're interested in mapping. So that primary axis of genetic variation through your entire data is correlated with many of the phenotypes. So here are the phenotypes that we've got, and those phenotype data come from the USDA GRIN database. This is over 15 years researchers are throwing data into this database. We don't know who collected the data. We don't, and sometimes they're collected in very different ways. It takes an enormous amount of curation time to go and grab the raw data from the USDA GRIN database and turn it into something that you're able to map with. But we went through this exercise, and then some of these I actually went out and measured them myself in the collection, so some of the size data and things like that. And what you see, what we've done here is comparing table and wine grapes, is there a significant difference for this phenotype? And if there is, you generally get a green arrow, and the direction of the arrow tells you where the increase is. So table grapes and eastern grapes are both firmer, longer, they have a more oblong shape, they're larger, they're heavier, and they have a larger width. But they're less dense clusters. That totally makes sense, right? Table grapes are big, and they're on those dangly clusters and so on. So we know that the phenotype data that we've got actually probably make good sense, but a lot of those phenotypes are very strongly correlated with population structure. So you've got rapid LD decay, you need 1 million SNPs to do GWAS. You've got traits that are correlated with population structure, intense relatedness among individuals, and we think, wow, let's give GWAS a try anyway, right? So let's see if it works. And when you run a genome-wide association study, you get one of these famous Manhattan plots. So just to go over this, you've probably seen them before, and if not, you'll see many again, I'm sure. So we've got all of the SNPs represented by a single dot here across the genome, and you've got chromosomes 1 through 19. You've got a random chromosome here of stuff that's unanchored to the genome. And the degree of association with the phenotype, in this case skin color that was scored from 1 to 5, is indicated along the y-axis as the minus log 10 p-value. So that a SNP up here with a value of 20 is a p-value of 1 times 10 to the minus 20. And in general, across these plots I'm going to show you today, the horizontal dotted line represents the significance threshold. Anything above that is suggestive that this might be biologically meaningful. So we take a look at this, and we see this huge peak on chromosome 2. So we also see this little helicopter over here. We get these all the time, and it's mostly due to the fact that the genome's just not put together very well. That SNP actually belongs on chromosome 2. It's just mismapped. And so there's lots of misassembly going into these, and so you need to be careful when you're seeing these helicopters floating around that you know, they don't necessarily mean that, oh look, it's 2 different genes controlling skin color. It's actually just one locus with a mismapped SNP. So this peak is very, very strong, and we have strong evidence that this is a real peak because the locus that controls berry color and grape is already known, and it's on chromosome 2, and it overlaps with this peak that we're finding. So if we zoom into chromosome 2 here, we find that mid-transcription factor is located here at the vertical dotted line, and the significant SNPs that we have kind of are scattered around that vertical dotted line. It's not that the SNPs that we've got land right on the locus. If we had better SNP density, we would probably find that, but we do get the hit in general. So how is it that we actually find an association signal for a trait when we think we need 1 million SNPs, but we've only got a couple thousand? Well, the reason is that there's evidence that this has been under very strong selection. So the lighter skin pigmentation in grapes, that lack of color, the ancestral state is a red grape, dark grape that produces dark juice. White wines are the derived state, and there's been intense selection for that lighter pigmentation berry. And that intense selection for lighter pigmentation has taken those alleles for light pigmentation, and they've risen more rapidly than you would expect at random. And when that happens, it drags along a very large haplotype with it. Recombination hasn't had enough time to break that down into small chunks because it's risen really rapidly. So it sits on a very long extended haplotype and has extended LD around the region. That can be measured using a statistic called the cross-population extended haplotype homozygosity test. So we compare the haplotype homozygosity in the white grapes compared to the red grapes across the genome. And we see if the values for the SNPs in this region are unusual. Do they sit on unusually long haplotypes given the frequency of the allele? And we find this horizontal dotted line right here indicates anything above it is within the top 1% of values genome-wide. And so we see that, yes, indeed, the SNPs in this region do tend to sit on these extended haplotypes. And we have evidence that there's selection for this lighter berry pigmentation. When you have selection for a trait and you're thinking that your SNP density probably isn't high enough given genome-wide levels of LD decay, you might just get lucky because the locus you're looking for actually has extended LD, and the SNP density required to map it might not be as high as other places in the genome. So we look at another trait here, muscat aroma, and if you've ever tried a muscat wine, muscat of Hamburg, muscat of Alexandria, muscat, petit vin blanc, there's all sorts of different muscat varieties. What we did, we just went through the collection and we just took every variety name that had the word muscat and scored it as a 1, and then scored everything else as 0, and ran a GWAS with it to see if we could pick up the muscat locus. And we find this peak on chromosome 5. Now, it does not reach the significance threshold, but we know already from the literature, because it's already been determined, that there is one non-synonymous substitution in this DXS gene at the top of the terpenoid synthesis pathway that's responsible for that muscat aroma. And that floral, tropical, next time you go and get a bottle of wine, go get a muscat. They're really fantastic. That floral aroma is controlled by that DSX. We map it successfully. Here's the vertical dotted line that indicates the position of that gene. And indeed, if we look at the signature of selection around that locus, there's also been there's evidence that there's been selection at the DXS gene as well for that muscat aroma. So this all makes sense. We've demonstrated that even with this very low SNP density array, with the data that we've got being historical data from a database that needed a lot of curation. So you're dealing with very suboptimal genotype data, suboptimal phenotype data, and we think, okay, how far can we stretch this? So with the reasoning that you can combine kind of selective sweep mapping together with association mapping, can we go look at another trait that we don't have a positive control for? We don't have one single large effect allele known from the literature. And so we decided to look at berry size. Under the assumption that, yeah, very likely berry size has been a target of selection. It is the thing that really differentiates table grapes from wine grapes. And larger berries, well known that that's what table grape breeders are going after. So we take a look at berry size and we find lots of sort of suggestive peaks, not far off from what we were seeing for the muscat kind of thing, but where to go? You know, is anything in here looking promising, and which ones should we focus on? So by taking a look at peaks in this plot that overlap with signatures of selection, we kind of narrow it down and say, well, under the assumption that this might be under selection, let's combine the selective sweep mapping. And the only one that we see here is really chromosome 11, that this peak right here, if you zoom in on chromosome 11, we see, and we overlay it with the selection signature, we've got around this locus right here, we've got the signature of selection and some, you know, suggestive association. And by combining these two things, we think, okay, this is a good place to look. So unfortunately, the story ends there. We go and we look under this locus and we don't find any good candidate genes, but you know, we publish it and off we go with the next project. But the exercise is useful because it suggests to people that when you're dealing with these data and you're going to have suboptimal data in these, especially in non-model organisms, and that you may not think that you've got the SNP density that suffices to move forward, but it's worth at least pushing through. So now let's switch over to the apple. We got 100 million pounds of apples grown in the Annapolis Valley in Nova Scotia every year. If you ever want to come up and have a nice visit, let me know. I'll show you around. So the same thing with the apple, extremely rapid LD decay. Now this is all from our GBS data now. When we started with our GBS data, we were getting 10,000 SNPs extracted from the data, and now with imputation and some better procedures, a little bit more sequence data, we're up at around 150,000 SNPs. And so that's the best thing about next-gen data. There's going to be a new method coming out in a couple of months, and then you can just go and take your existing data and extract more information from it. But you've got this very rapid LD decay in apple, also same situation. Any two SNPs within 500 base pairs are often not even correlated with one another. You need millions of SNPs to go across the genome. I should have mentioned both grape and apple are diploid. Grape has a genome size pretty compact, around 500 megabases, and apple's about 750 megabases. So same thing with apples also in terms of relatedness among individuals. If you look at the top 14 apple varieties, this is also data from the USDA apple germplasm collection, which is up in Geneva, New York. And you take a look at the top 14 varieties in our data in terms of acreage across the US. You see that on many of them, the ones in red, they're connected to other varieties here by a first-degree relationship. You see Macintosh, very heavy there. So what happens is you get a winner for a cultivar, and breeders reuse them as parents, and then you end up with this really extensive sort of relatedness network. What's interesting is that Honeycrisp is out here on the end here, probably one of the most successful commercial cultivars in the store right now that has very little relatedness. In fact, it was a like a miscross, sort of unknown pollen parent floating in kind of thing, which is one of the reasons why I thought it'd be good to get into apple, since the most popular commercial cultivar wasn't actually an intentional cross. I thought maybe we could do a little bit better than random. But you see this intense relatedness again in the apple. And when we look at patterns of population structure using principal components analysis, same sort of deal. What we see is in the grape, the main axis of variation here, PC1, tends to be associated most strongly with the harvest date. So early varieties tend to be over here on the left-hand side of the plot, while later varieties tend to be over here in blue. And we've also got New World and Old World apples, so Old World being from Europe primarily, and then the New World apples being from the US, that generally the circles tend to be down here on PC2, and the triangles up here on PC2. So we see that the axes of variation tend to be also correlated with geography, if you will, or with whether you're an early or a late apple. And so that's important because early apples actually tend to be less firm, and late apples tend to be firmer, that we'll see here in a moment. So let's run a GWAS with our GBS data and see if we can find anything. And let's start with skin color because it's really easy. And we score skin color. We take it out of the GRIN USDA database, link it up with all the GBS data, run a genome-wide association study, and we find this big hit on chromosome 9. Again, you've got a couple Hell helicopters that you can safely ignore. It is one single locus of large effect on chromosome 9, another MIB transcription factor, and there's a huge signature of selection here for red apples. It turns out that there's been very intense selection to try to generate apples that are redder and redder and redder. And so we pick up that signature of selection in the genome. So now moving on to some traits that we looked at. These are populations up in Nova Scotia or the USDA. There's sort of a mishmash of data here, but we've got harvest date. So harvest date, we get two very big hits on chromosome 3 for harvest date, and for firmness as well, the exact same two SNPs here. That top SNP up here turns out that no one's really mapped this location before, and when we look at it in more detail, that top SNP actually is a non-synonymous substitution in the middle of a NAC transcription factor. Those NAC genes are known to be involved in ripening in banana and in kiwi, and the NOR in tomato is the ortholog in tomato. We know that it's an excellent candidate gene. And there's a non-synonymous substitution at the 5th amino acid that happens to be that SNP that we genotyped. It doesn't mean that this is the causal allele. It just means, but it's not a bad candidate since it is a non-synonymous substitution in the middle of a transcription factor involved in ripening. So we figured we were on the right track. You can see the multiple species alignment here, that the ancestral allele, D, is really quite conserved across a lot of species. And the switch to the Y here is actually the derived Y actually creates an apple that is less firm. So we figure that it's actually a gain of function mutation that actually accelerates ripening. But we like to be able to check these things out and look to see whether that association holds in other populations. So we got some Danish collaborators that did it, and we find the same two hits for an association with firmness on chromosome 3. And then we do it in a population in Nova Scotia where the top hit is the exact same SNP. Doesn't reach significance threshold, but it's still the same SNP. Then we do it in another population in Nova Scotia, and we find the exact same hit. So we've done it in several different populations now, and it seems very robust that this is likely the largest effect allele controlling firmness in apple. And it's never been described before. And what's particularly surprising is that there are 3 markers out there that are routinely used in marker-assisted breeding in apple that are supposed to be predictive of firmness. But they do not overlap with the main locus that we find doing GWAS. So the reason for this is that all of this mapping that's been done to find these loci has been done in biparental linkage mapping populations. It's not unlikely that some of these loci actually do control firmness. They might be low frequency, large effect alleles that segregate in those parents, but they don't necessarily transfer to diverse germplasm. So when you're looking at a large population of diverse material, these alleles are not predictive of firmness, and we've actually gone in and genotyped those exact markers that are used in marker-assisted breeding, and they do not show up as being significant here. But what is interesting about PG1, this is a polygalactoronase, probably the most well-studied one, and there's good evidence that it should be involved in ripening since the gene is known to be involved in ripening, and it's been studied quite extensively. What we did is we did an experiment where we did get a hit on chromosome 10 that looked like it was very close to the polygalactoronase gene, but it was for change in firmness, not just firmness at harvest. Okay? So firmness at harvest, we measure, we have an automated penetrometer, goes in, measures the firmness of the apple, and we do half the apples in the bin, and then we take the rest of the bin, we put it into storage, we take it out 2 months later, we put it back through, and then we look at the difference in firmness and how much firmness loss there has been. And when we map that, we find that it overlaps on chromosome 10 very close to that polygalactoronase gene. We think, hey, we got PG1. Looks like PG1's involved in the loss of firmness, not in just firmness. However, when we look at it in detail and we zoom in on that region, we find actually the PG1 mutation is right here. It's not significantly associated with change in firmness. And it's 500 kb away. It's a 500,000 nucleotides away from the actual top SNP that we've got, which overlaps really well with an ethylene response factor, right? So what this is telling you is that when you're using genome-wide association, you've got much better resolution than you do in these linkage mapping populations in a long-lived woody perennial that takes 6 years to grow. Like, you're never gonna have a population of 2,000 plants here. We're talking about linkage mapping in biparental crosses where both parents are heterozygous with like 200 or 300 offspring tops. And so your resolution is really poor, so that you get these huge regions and then you look inside and they go, PG1. That looks like a good candidate gene. That's our marker. Let's go with it. PG1, PG1. But not until you actually do some GWAS and be able to narrow that down and get better resolution do you realize that they may have been a little bit off target, and that it's more likely now that variation within and around that ERF is responsible for the loss in firmness phenotype. That doesn't mean that all the markers they're using in marker-assisted breeding don't work. The MA1 for acidity, when we run acidity and we genotype MA1, we get the hit, but MA1, the actual marker they're using is the top hit, and so it seems like that one works quite well in diverse material. So with this all said, it seems to me that in log-lived woody perennials like apples and grapes, it's a great idea to go and put your populations together now. The sooner the better. If you work on almond, if you work on walnut, if you work on whatever, go put a big association mapping population in the ground. We did this 6 years ago when I started my position in Nova Scotia. We imported the entire USDA collection into Nova Scotia and then added a bunch of varieties from advanced breeding lines from the Canadian Breeding Program, and put together over 1,000 varieties that we've planted in duplicate on the same rootstock in a controlled design with checks and the whole deal, all barcoded up, ready to go. And we're very focused now on high throughput phenotyping, where we have a machine that can take, we did about 16 or 17,000 apples this year that we put through this machine that has an automated penetrometer, takes the weight, smushes the apple up, it does the sweetness, the acidity. It measures the weight of the pulp and the juice and gives you juiciness measures, and it does that all in an automated fashion without ever having to write anything down or anything like that. We also got a new machine that we call the Snapple that takes snapshots of every apple, and so when you put it through, it'll take a picture on the side and the top there, and we can start to do image analysis of shape and size and color. So ultimately, I'm not going to get into what I'm really interested in doing, because now you can see, you're 6 years in. I've got this population in the ground in Nova Scotia, which I consider a public resource. So if anybody ever wants thinking that they'd like to go and phenotype an apple population and link it up with some genotype data, then let me know. But the next step really is, how do I combine all these things and pyramid them in so we get an apple that has all of those quality traits that we're interested in. We don't do a lot of disease resistance work, obviously, because you can't inoculate the entire collection and kill it. But those traits also have been mapped by other groups, and piling those all in to make an apple that really contains all the traits you're interested in. That's the next steps that our lab is interested in taking, and that's why I'd like to hang out in UC Davis next year a little bit and figure out how that gets done. So thank you very much for your time, and if you have any questions, please let me know. Go ahead. Yeah, so do we end up missing genes? For sure. Yep. 30% of the apple genome isn't even assembled. So yeah, we're missing like 30% of the genes already without even... And for grape, I don't know what the assembly is on, the new assembly on grape, I don't know how complete it is. But yeah, inevitably, without complete genome saturation with markers, you're going to be missing things. And right now, you can see we're really going after the lowest hanging fruit, right? We're going for single effect alleles, controlling traits that are easy to measure, many of which have been targeted by selection, you know, but when we want to get into traits that have not been targeted by selection, where LD breaks down more rapidly and that are controlled by a large number of small effect alleles, that's going to be more difficult. Of course, I didn't talk anything about genomic selection, but obviously that plays a role as well for many of those traits, and we've done some work on that. Yeah, it's an excellent question. How many candidate genes does there end up being when you get a hit like that? You know, a lot of people, you look in the literature and everybody does it a little different. I'll go 10 kb out this way and 10 kb out that way, grab all the gene names, stick them in the supplement, go publish the paper, and move to the next project, right? And so often for us, I find that's the most difficult part of my job because I'm not really a real biologist. Just do the gene hunting. And so then you go and you publish it, and then you move to the next project and go sequence some cannabis or something. But I think that's where I think it gets interesting, is to try to figure out which are the best candidates and how far out do you go from that locus. You know, you take a look at the patterns of LD and you can make some guesses, but it is a tricky thing to do. You know. Go ahead, Molly.
Long haplotypes.
Pardon me?
You were talking about longer haplotypes in grapes that you have seen in the germplasm collection.
Yeah.
Germplasm collection usually represents historical recombination which has occurred. If they have remained as a one haploblock, what does that mean evolutionarily? Do you think you will get a recombination in that region, or is it located somewhere around the centromere where recombination is very rare?
I think we can get recombination if people start making some more crosses, which would be great. But yeah, I mean, the larger the diversity that you're sampling, the more likely you are to have those haplotypes broken up. So it's really a matter of sample size a lot of the time to be able to try to find, you know, around those, especially around those selected loci where you have long haplotypes, where it gets broken up. It's a matter of, you know, the only thing we can really do is just keep sampling as many individuals as we can, hope to get more recombination events.
So you mean that recombination is possible in that haploblock?
Yeah.
If we increase the sample size.
Any other questions?
Yes, you seem to dismiss the role of biparental crosses, or maybe I got that wrong. Or
that's because I have a big GWAS population.
Yeah, because I thought that the example you gave, but would be valid for other traits, is that precisely, you know, you dismiss these other markers because they appear to be low frequency in your population, or GWAS population. But from a breeding standpoint, wouldn't these other markers be useful if you're trying to accumulate genes that control the same trait, and you want to maximize expression? And so, you know, I think it is dangerous to just rely on GWAS.
Yeah, no, for sure. And I don't want to dismiss the results from the linkage mapping populations. The lesson really is that those markers may be useful in those biparental crosses, in closely related material. But when you look at diverse material and see whether they're actually accounting for phenotypic variation, they don't. And so that they're complementary, they're really complementary approaches.
But I think that, you know, the height of the peak measures the confidence that, it has a major effect, or it has high frequency in your population. It does not measure the usefulness in breeding, actually.
That's right.
Obviously.
Yeah. And you know, the reason why we don't get an association at those other three markers is not necessarily because of frequency. So that's the other thing. PG1's actually, the alternative allele is actually quite common in our population, yet it still doesn't associate with firmness. So, which, you know, in talking to the people who discover PG1, find extremely surprising. They think, no, it is the thing that controls firmness in apples, right? In their collections of biparental crosses and in their breeding program. The problem is that right now in the Canadian breeding program, we're using those markers to screen offspring, and those offspring are not generated from the same genetic material that the discovery was made in, and it's unlikely that they're actually accounting for any variation in the offspring, but we're culling offspring using these markers when perhaps we shouldn't be. If I were a geneticist and a breeder, I would start with NAC. I wouldn't start with PG1 in my population to be screening. So. Anything
Okay,
else?
thank you.
Great. Thank you very much.